All right, listener, get ready, because today we're going deep into the world of protein synthesis.
Ooh, exciting.
Yeah, it's complex, lots of moving parts,
Uh,
but don't worry, I think our listeners are definitely up for the challenge. We're
Absolutely.
going to break it all down,
Yeah.
all those steps, the molecules involved, see how it all comes together to make, the essential building blocks of life.
The elegance, the precision, the efficiency of it all is it's amazing, really, when you consider it. Evolution honed these processes over billions of years.
So we're going to dive straight into the details here.
Okay.
Assume you know the basics, right? We're not going to rehash those. We're diving straight in, starting with prokaryotic elongation.
Excellent point. We're focusing on the bacteria first.
Yeah.
Prokaryotes and eukaryotes have their similarities and differences, but
Yeah,
for now, let's just stick with
Okay.
how bacteria get it done.
So when we talk about prokaryotic elongation, this is where the polypeptide chain, really starts to grow, right?
Yeah. Exactly. We're way past initiation now. Ribosome's all set, ready to add amino acids one by one.
Mm-hmm.
And it's all according to those instructions that are encoded in the messenger RNA.
So what's the first step in this elongation dance? What happens?
Okay, so first, the second amino acyl tRNA, bind that to the ribosome.
Okay.
Remember the initiator tRNA
Yeah.
carrying formylmethionine?
Right.
That's already bound to the P site.
Okay.
Now got to bring in the next amino acid.
So that's where our good friend elongation factor 2 comes in. EF2.
Precisely.
Okay.
EF2, it's a GT-PACE, meaning it binds to GTP and it hydrolyzes it to GDP
Right.
in its GTP-bound form.
Okay.
That's when it exports the amino acyl tRNA to the ribosome.
So it's kind of like a chaperone, making
Mhm.
sure it gets to the right place.
That's a really good analogy, yeah. And
That's
it's
pretty-
very specific. EF2
Yeah.
only binds to amino acyl tRNAs, not to uncharged tRNAs.
Oh, okay.
So that makes sure that only the right amino acid gets delivered to the ribosome.
So it brings it to the ribosome. Where does it bind specifically?
It binds to the A site,
Okay.
which stands for amino acyl site.
Oh, okay.
That's where the incoming amino acyl tRNA, the one carrying the next amino acid to be added, that's where it binds.
So we've got the initiator tRNA in the P site,
Yep.
and the second amino acyl tRNA is now in the A site.
Yes.
What happens next?
Peptide bond formation between the two amino acids.
Okay.
Now this reaction, it's catalyzed by peptidyl transferase.
Peptidyl transferase.
Yeah,
Big word.
it is.
What does it do?
Okay, so it's a ribozyme,
Oh.
meaning it's an RNA molecule,
Mm-hmm.
but it has catalytic activity. It's
Right.
actually part of the large ribosomal subunit.
Okay.
And what it does is it facilitates the transfer of the growing polypeptide chain from the tRNA that's in the P site
Mm-hmm.
over to the amino acyl tRNA in the A site.
So it's like pass from one tRNA to the other.
Yes, exactly.
That's really interesting.
Yeah.
Where does the energy for this reaction come from?
Okay, so the energy actually comes from the high energy bond that's between the amino acid and the tRNA in the P site.
Mm-hmm.
This bond was formed earlier
Okay.
during amino acid activation.
Right.
We won't get into the details of that right now.
Okay.
It's another important part of this whole protein synthesis process.
So we've got the peptide bond formed. Now what?
Now we're going to move the ribosome along the mRNA
Okay.
so we can add the next amino acid.
Right.
This is called translocation,
Mm-hmm.
and that's facilitated by another elongation factor
And there's another one.
called EFG.
EFG. Okay.
Mm-hmm. Another elongation factor.
Lots of players in this dance, huh?
Yeah, there are a lot, but each one plays a very specific role.
Okay.
So EFG, also a GT-PACE,
Okay.
uses energy from GTP hydrolysis
Mm-hmm.
to move the ribosome one codon down the mRNA.
So it's kind of like the engine that drives the ribosome along.
That's a good way to think about it, yeah. As the ribosome moves, the tRNA that was in the P site, now carrying the empty tRNA, it shifts to the E site,
Okay.
which is the exit site.
Okay.
The tRNA that was in the A site, now carrying that growing polypeptide chain, it moves to the P site.
Right. Right.
And now the A site, it's empty, ready to receive the next amino acyl tRNA.
It's like a well choreographed little ballet.
Exactly.
All these tRNAs moving from one site to the next, and this polypeptide chain just keeps growing and growing.
Yeah, and the cycle, it just repeats over and over again. each cycle adding one amino acid to that polypeptide chain.
It's amazing how efficient it is, but it must take a lot of energy, right?
Oh, it does. Each cycle requires the hydrolysis of two GTP molecules,
Wow.
one by EF2 and one by EFG.
Okay.
And remember, that's just for adding one amino acid.
Right.
So think about the energy for synthesizing a protein that's hundreds or thousands of amino acids long.
Mind-blowing. Worth it, though, because proteins are so important.
Oh, absolutely. They're essential for virtually every aspect of life. Workhorses of the cell carrying out such a wide range of functions.
Now, this elongation, it keeps going and going until the ribosome hits a stop codon, right?
That's right. When a stop codon enters the A site, there's no corresponding tRNA to bind to it.
Okay.
So instead, a protein called a release factor
Okay.
binds to the ribosome.
So it's like, hey, we're done here.
Exactly. The release factor triggers hydrolysis of the bond between the polypeptide chain and that tRNA in the P site,
Okay.
releasing that completed protein.
And what happens to the ribosome after it releases the protein? Does it just, like, fall apart?
Not quite, no. It undergoes something called ribosome recycling.
Okay.
It gets disassembled into its subunits, the small subunit and the large subunit.
Okay.
And then those subunits can be used to initiate translation of another mRNA molecule.
Oh, so they just get reused. That's fascinating.
Yeah, it's very efficient.
So in prokaryotes, transcription and translation, that's coupled, right?
Yes, that's a key difference. Okay.
Meaning that ribosomes can begin translating an mRNA molecule, even while it's still being transcribed from DNA.
So it's like multitasking.
Precisely. And this coupling allows for very rapid protein synthesis,
Oh, wow.
which is essential, for that rapid growth and adaptation that bacteria are known for.
Fascinating. All right, so we've covered a lot here, just about prokaryotic elongation. That was a lot. But it's just like a crucial part to understand, to grasp the whole beauty and elegance of protein synthesis.
Absolutely.
All right, listener, we've gone through the steps of prokaryotic elongation, from the binding of the second aminoacyl tRNA to the ribosome, to the formation of the peptide bond, to the translocation of the ribosome, and finally to the release of that completed protein.
Yeah, we've talked about those key players involved, the elongation factors, EF2 and EFG,
Right.
the ribozyme peptidyl transferase, and the release factors. Even touched on the energy requirements, the efficiency of the cell, and recycling all this molecular machinery. So hopefully you're starting to see how this whole intricate dance of molecules unfolds within the ribosome, right?
Yeah.
This incredible precision of protein synthesis.
It's amazing.
But we're not done yet. There's a whole other world to explore.
Oh, yes.
Eukaryotic protein synthesis.
Can't wait to dive into
At least
that.
ready for that.
Absolutely.
Eukaryotic protein synthesis, it has similarities to what we just talked about,
Right.
but it also has its own unique features and, honestly, even more complexities.
Yeah. A lot more complex.
I'm sure our listeners are ready to learn more about that.
Absolutely. Let's dive into that world, eukaryotic protein synthesis.
Okay.
Now, it reflects the complexity of those eukaryotic cells. It's a lot more intricate.
Right, because they've got that nucleus,
Exactly.
which means transcription and translation, those are separated
Mm-hmm.
in space and time, unlike in prokaryotes where it's like happening simultaneously.
Exactly. So in eukaryotes, transcription, that happens in the nucleus where DNA is transcribed into messenger RNA or mRNA.
Okay.
And that mRNA needs to be processed,
Okay.
transported out of the nucleus into the cytoplasm. That's where translation takes place.
So it's like an extra layer of complexity
It is.
compared to prokaryotes.
Right.
But this separation, it allows for more sophisticated regulation of gene expression, right?
Exactly. Yeah.
So how does the mRNA get processed in the nucleus? Like before it's shipped out to the cytoplasm, what happens?
Okay. So there are three main processing steps. First, a 5' cap is added to the beginning of that mRNA molecule.
Okay.
Now, this cap, it's a modified guanine nucleotide,
Mm-hmm.
and it protects the mRNA from degradation,
Okay.
and it helps it bind to the ribosome during translation.
So it's like putting a helmet on it,
Yeah,
protecting it before it goes out into the cytoplasm.
a good analogy.
Yeah.
Second, a poly-A tail
Mm.
gets added to the end of that mRNA molecule.
Okay.
Now, this tail, it's a string of adenine nucleotides,
Mm-hmm.
and it also helps protect the mRNA from degradation. and it signals that it's ready for export from the nucleus.
So it's like a little flag that says, get me out of here.
Exactly, yeah. And then finally, the mRNA undergoes splicing, where those non-coding regions called introns are removed.
Okay.
And the coding regions, exons, are joined together.
Splicing sounds like a very delicate operation. Why does it need to be spliced?
Well, the introns, they don't contain instructions for making proteins.
Okay.
So they need to be removed before the mRNA can be translated.
Right.
So splicing ensures that only those coding sequences are included in the mature mRNA molecule that's exported to the cytoplasm.
So now the mRNA, it's been capped, it's been tailed, and it's been spliced.
Exactly.
All dressed up, ready to go to the cytoplasm. What happens next?
Now, that mature mRNA molecule is transported out of the nucleus through nuclear pores into the cytoplasm, and there it encounters ribosomes, the protein synthesis machinery.
Eukaryotic ribosomes, those are different from prokaryotic ribosomes, right?
Yeah, they are.
In what way?
Well, they're larger, more complex,
Okay.
made up of different ribosomal RNAs and proteins. They're also found in different locations within the cell.
Okay.
Some are free-floating in the cytoplasm.
Right.
Others are attached to the endoplasmic reticulum, or ER, and the location of the ribosome.
Yeah.
That determines where the protein will end up.
Oh, okay.
So proteins synthesized by ribosomes attached to the ER, those are typically destined for secretion outside the cell or for insertion into cell membranes.
Got it.
But those proteins that are synthesized by free-floating ribosomes, they usually function within the cytoplasm.
Okay, so let's zoom in on that translation process at the ribosome. How does it differ in eukaryotes compared to what we just discussed for prokaryotes?
Okay, so the basic steps of elongation and termination, those are similar in both prokaryotes and eukaryotes. You still have the binding of aminoacyl tRNAs to the A site, the formation of the peptide bond, the translocation of the ribosome, and the release of that completed protein. But there are some key differences in the initiation stage.
Okay, let's talk about those differences. What's different about initiation in eukaryotes?
First, the initiator tRNA in eukaryotes carries a regular methionine.
Okay.
Not a formal methionine like in prokaryotes.
Right.
Second, the initiation process, it involves a much more complex set of initiation factors called EIFs.
Okay.
There are over a dozen EIFs.
Over a dozen.
Each playing a specific role in assembling that initiation complex.
Wow, a lot of factors. What do they all do?
They have various roles, like recognizing the 5' cap of the mRNA, recruiting the small ribosomal subunit, bringing in the initiator tRNA, and scanning the mRNA for the start codon.
Scanning the mRNA for the start codon. That's interesting.
Yeah, so in eukaryotes, the start codon, it's not always the first AUG codon in that mRNA sequence.
Yeah.
So the ribosome, it needs to scan the mRNA to find the correct AUG codon.
Right.
That signals the beginning of the protein coding sequence.
So it's like double checking, making sure it's starting in the right spot.
Exactly. It's really crucial for making sure that translation starts at the right place.
So the initiation complex is assembled, start codon's been located, the ribosome's ready to go. What happens next?
Now we enter the elongation phase, which, as I mentioned, is similar in principle to prokaryotic elongation.
Okay.
You have EF2 bringing in aminoacyl tRNAs to the A site, peptidyl transferase catalyzing the peptide bond formation, EFG driving translocation of the ribosome.
So it's the same basic steps, just different players.
Yeah, you can think of it that way. They have different names, but the functions are analogous.
Okay, and termination. Does that follow the same basic principles with like release factors recognizing the stop codon?
Exactly.
Triggering the release of the completed protein?
Yep. The eukaryotic release factors are different from the prokaryotic ones, but they do the same essential function.
Okay, so we've talked about the basics of eukaryotic protein synthesis, but I feel like there's got to be more to the story.
Oh, there is.
What about all those post-translational modifications we talked about earlier?
Ah, yes. Post-translational modifications. Crucial part of eukaryotic protein synthesis, adds another layer of complexity and regulation.
Right. Can you remind me what those are all about?
Sure. So these are chemical changes that are made to a protein after it's been synthesized.
Okay.
These modifications can alter the protein's structure, its function, its location, or its stability.
So it's like accessorizing, like taking a basic outfit and making it more stylish and functional.
That's a great way to think about it. And there's a vast array of post-translational modifications, each one with its own specific purpose.
Give me some examples.
Okay. Phosphorylation, glycosylation, lipid modifications, ubiquitination, disulfide bond formation, and many more.
Wow.
Each one adds a unique chemical group to the protein, altering its properties in specific ways.
So it's like a molecular toolbox for customizing proteins.
It is.
That's really cool. Can you break down a few of those for me?
Sure. Let's start with phosphorylation.
Okay.
One of the most common post-translational modifications
Okay.
involves adding a phosphate group to specific amino acids, typically serine, threonine, or tyrosine.
Okay. And what does adding a phosphate group do?
Well, it can act like a molecular switch, turning a protein on or off. It can change a protein's shape, its interactions with other molecules, or its location within the cell.
Wow. So it's a very powerful way to regulate protein activity.
It is.
Wow.
Phosphorylation is involved in so many cellular processes: cell signaling, metabolism, gene expression.
What about glycosylation? What's that all about?
Glycosylation involves adding sugar molecules, or glycans, to proteins.
So it's like decorating a cake.
Yeah, kind of like that.
Yeah.
These glycans can influence protein folding, stability, and interactions with other molecules.
So they're not just decorative?
No, they serve an important purpose.
No.
There are two main types of glycosylation: N-linked and O-linked. N-linked glycosylation happens in the endoplasmic reticulum, or ER. O-linked glycosylation occurs in the Golgi apparatus.
So it's like the protein goes through different finishing departments within the cell.
Exactly, and each department adds its own specific type of glycosylation, customizing the protein's properties even further.
That's cool. What about lipid modifications? Does that like involve attaching fat to the protein?
Yes, exactly.
Yeah.
Lipid modifications involve adding fatty acid chains to the protein, typically to cysteine residues.
Okay, and why would a protein need like a fatty acid makeover?
Well, those fatty acids can act as anchors,
Okay.
helping the protein insert itself into cell membranes.
Ah, so it's like giving the protein a little hydrophobic tail
Yeah.
so it can hang out with the lipids in the membrane.
Exactly.
Okay.
It's all about targeting that protein to its proper location.
Got it. What about ubiquitination? That one sounds a little ominous.
Ubiquitination is a fascinating process. It involves attaching a small protein called ubiquitin to other proteins,
Yeah.
and it often acts as a signal for protein degradation.
So like a kiss of death.
Yeah, you could say that. Proteins that are tagged with ubiquitin are typically targeted for destruction by the proteasome,
Okay.
which is a cellular machine that's responsible for breaking down proteins.
So ubiquitination is a way for the cell to get rid of like old or damaged protein.
Exactly.
Like a garbage disposal.
Exactly.
So-
Crucial for maintaining cellular health
Right.
and removing proteins that are no longer needed or potentially harmful.
And finally, we have disulfide bond formation. Is that like the bonds we learned about in chemistry class?
Yeah, it is. Disulfide bonds are covalent bonds formed between two cysteine residues.
Okay.
They're like tiny little bridges that help stabilize protein structure.
So they're like the scaffolding that holds the protein's shape.
Exactly.
Okay.
And they're particularly important for proteins that function in really harsh environments like the extracellular space.
It's amazing how all these modifications work together to like fine-tune protein function, make sure that they're properly localized and stable. It's like a complex choreography,
It really is.
with each step playing a crucial role in that final performance.
It's a testament to the complexity and the elegance of cellular processes, but to really appreciate the impact of post-translational modifications.
Yeah.
Let's look at a specific example.
Okay, I'm all ears.
Okay, let's consider the modification pathway of insulin.
Insulin, okay.
It's a classic example of how post-translational modifications transform a precursor protein into a mature functional hormone.
Yeah, insulin. That's the hormone that regulates blood sugar levels.
Exactly.
Yeah, that's a familiar one.
So insulin starts its life as a single polypeptide chain called preproinsulin,
Pre-proinsulin, okay.
synthesized in the beta cells of the pancreas.
Mm-hmm.
And right after synthesis, it's translocated into the endoplasmic reticulum, or ER.
So again, it's like sent off to a finishing school within the cell.
Exactly. The first step in this finishing school is the removal of the signal peptide.
Signal peptide, okay.
A short sequence of amino acids
Okay.
directs the protein to the ER. This cleavage converts preproinsulin to proinsulin.
So the signal peptide is like the protein's address label,
Yeah,
guiding it to the right location, and then once it arrives, the label is removed.
exactly.
Okay.
Now, inside the ER, proinsulin undergoes a crucial step, folding. It adopts a specific three-dimensional structure
Right.
essential for its function.
Okay.
During this folding process, disulfide bonds are formed, further stabilize that protein structure.
So it's not just a linear chain anymore. It's folding up into a specific shape, held together by those disulfide bridges.
Precisely. And once proinsulin is properly folded,
Mm-hmm.
those disulfide bonds are in place, it's transported to the Golgi apparatus, the next finishing department in our analogy.
What happens in the Golgi? More modifications.
Yes, definitely.
Okay.
In the Golgi, proinsulin is packaged into secretory vesicles, tiny little membrane-bound sacs.
Okay.
They'll eventually release insulin into the bloodstream. But before that can happen,
Yeah.
proinsulin needs to undergo another crucial modification.
Okay. What happens now?
Okay, so inside these secretory vesicles, a specific enzyme cleaves proinsulin, removing a section called the C-peptide. This cleavage generates two chains,
Mm-hmm.
the A chain and the B chain,
Mm-hmm.
which are held together by those disulfide bonds we talked about.
Right.
So now we have two chains instead of one.
Mm-hmm.
These are the chains that make up mature insulin, right? Almost. There's one final step. Another enzyme, carboxypeptidase E, trims a few amino acids from the end of the B chain.
Okay.
And voila, mature functional insulin.
It's amazing how much processing goes into creating this tiny hormone. So we go from pre-proinsulin to proinsulin. Cleavage, trimming, and finally mature insulin.
Yeah, it highlights the importance of these post-translational modifications in creating those functional proteins.
Yeah.
They're not just add-ons.
Right.
They're essential for proper protein function, localization, regulation.
It's a good reminder that protein synthesis isn't just stringing amino acids together. It's a very complex, multi-step process with all these intricate checks and balances, modifications that fine-tune the final product.
Absolutely. And each of these modifications adds another layer of complexity to the already fascinating world of protein synthesis. But let's not forget those individual amino acids.
Right.
They are the building blocks of proteins.
Yeah.
And they can undergo their own set of modifications as well.
Right. Like we talked about phosphorylation earlier.
Yeah.
But what other modifications can happen to individual amino acids?
Okay, so you've got methylation, carboxylation, hydroxylation, and more. Each modification alters the chemical properties of that amino acid,
Mm-hmm.
which in turn can influence protein function.
Okay, let's break those down one by one. What about methylation? What's that?
Methylation involves adding a methyl group,
Okay.
a carbon atom with 3 hydrogen atoms,
Mm-hmm.
to an amino acid.
And which amino acids typically get methylated?
Lysine is a common target. It can be methylated once, twice, or even 3 times.
Wow.
Each level having a distinct impact on protein function. And there are specific enzymes that are responsible for each step of methylation,
Right.
adding another layer of regulation.
So it's like a little fine-tuning knob,
Yes,
adjusting the protein's activity with each additional methyl group.
exactly. Methylation, it plays crucial roles in various cellular processes, from DNA replication and repair to gene expression and signal transduction.
Wow. Okay, what about carboxylation? What happens there?
Carboxylation involves adding a carboxyl group, a carbon atom double bonded to an oxygen atom,
Okay.
and also bonded to a hydroxyl group.
Mm-hmm.
Glutamate, or glue, is a common target. Carboxylation can change a protein's charge and its ability to interact with other molecules,
Okay.
influencing its function.
So like adding a little sticky note to the protein,
Yeah.
changing how it interacts with its surroundings.
Good analogy.
Okay.
Carboxylation is involved in various processes: blood clotting, bone formation.
Okay, what about hydroxylation? Does that involve adding like a water molecule to the amino acid?
Close. Hydroxylation involves adding a hydroxyl group,
Right.
an oxygen atom bonded to a hydrogen atom.
Hmm.
Proline, or pro, is a prime example. And the hydroxylation of proline, that plays a crucial role in the formation of collagen, a fibrous protein that provides structural support to our tissues.
Collagen, right? Like keeps our skin firm and our joints flexible? That's
Exactly.
an important one.
It is. The hydroxylation of praline to 4-hydroxyproline, or 4-HEP, is essential for the proper assembly of those collagen fibers.
So without that hydroxylation, our collagen would be like floppy and weak.
Exactly. And vitamin C actually plays a crucial role in this process.
Oh, wow.
It promotes the activity of the enzymes responsible for proline hydroxylation. That's why vitamin C deficiency can lead to scurvy, that condition characterized by weak connective tissues.
Wow. So these tiny modifications can have a big impact on our health.
They do. Really underscores the importance of understanding all these details of protein synthesis and modification.
Yeah, absolutely. Okay, let's zoom in on a specific example of uh, how methylation can influence protein function.
Sure.
Like a real specific example.
Okay, let's talk about histones.
Histones, okay.
They're proteins that package and organize DNA within the nucleus.
Right, those little spools that DNA wraps itself around.
Exactly. And the methylation of histones plays a crucial role in regulating gene expression. It's part of a field called epigenetics, which studies heritable changes in gene expression that don't involve alterations to that underlying DNA sequence.
So it's like a layer of control on top of the genetic code?
Precisely.
Wow.
And methylation, it can either activate or silence genes depending on which histone and which amino acid is methylated.
Wow, okay.
The methylation of lysine 9 on histone H3, or H3K9 for short,
Okay.
that's associated with gene silencing.
Okay.
It promotes the formation of heterochromatin, tightly packed form of DNA. That's transcriptionally inactive.
So it's like putting a padlock on a gene, preventing it from being expressed.
Exactly.
Okay.
There's a protein called HP1.
Okay.
It recognizes and binds to methylated H3K9, further promotes chromatin compaction and gene silencing.
So it's like a security guard,
Yeah,
enforcing that lockdown on the silenced gene.
exactly. Now, on the other hand, the methylation of lysine 4 on stetone H3, or H3K4,
Okay.
that's associated with gene activation,
Okay.
promotes the formation of euchromatin, a more relaxed form of DNA. That's accessible for transcription.
So like opening up the book of DNA so the cell can actually read those genes within.
Exactly. Methylation of H3K4 creates a more permissive environment for gene expression.
So these tiny methyl groups can have a huge impact on gene expression.
Yeah.
It's like really highlights the complexity and the subtlety of epigenetic regulation.
It does.
Wow.
Underscores the fact that inheritance is not solely determined by our DNA sequence.
Right.
Those epigenetic modifications that decorate our DNA,
Yeah.
those can also be passed down from one generation to the next, influencing the traits we inherit.
So like a whole hidden layer of information
Yeah.
on top of the genetic code shaping who we are.
Exactly. Epigenetics is a rapidly evolving field with profound implications for our understanding of health, disease, even evolution.
Okay, let's move on to another intriguing modification. N-linked glycosylation.
Okay.
And I touched upon it earlier, but let's get into the details.
Okay, so N-linked glycosylation involves adding a complex sugar molecule, or glycan, to the nitrogen atom of an asparagine residue,
Okay.
or acin in a protein.
And this happens in the ER, right?
Correct.
Okay.
Process starts with a special lipid molecule called dolcyl phosphate, acting as a carrier. It picks up sugar molecules one by one, building a chain of sugars.
So it's like a construction worker.
Yeah.
Building a little sugary scaffold.
Good analogy.
Okay.
The first two sugars added are N-acetylglucosamines, or GLC-NS. Then 5 mannosugars, or MAN, are added, forming a 7-sugar backbone.
Okay, so we've got this sugary backbone attached to the liquid phosphate. What happens next?
Now comes the flip.
The flip.
Yeah. This entire sugar chain, still attached to dolichal phosphate, gets flipped from the cytoplasmic side of the ER membrane to the luminal side, the space inside the ER.
So it flips over the fence into the ER's like inner sanctum.
Exactly.
Okay.
And once inside the ER lumen, that sugar chain, it gets further elongated. 4 more mannosugars and 3 glucose sugars, or GLC, are added.
So the sugary scaffold's getting bigger and more elaborate. What's the purpose of all these sugars?
Well, they play crucial roles in protein folding, stability, interactions with other molecules.
But how does this sugar chain actually get attached to the protein?
That's the final step.
Okay.
The entire sugar chain, still attached to dolichal phosphate, is transferred en bloc to a specific asparagine residue, acinin, on the protein.
So it's like the construction worker finally attaches this elaborate sugary chandelier to the protein ceiling.
Precisely. And once that sugar chain is transferred,
Yep.
dolichal phosphate, it's recycled.
Oh, okay.
Loses a phosphate group, flips back to the cytoplasmic side of the ER membrane, ready to pick up more sugars, start the process again.
It's like a perfectly choreographed little dance.
It is.
Each molecule playing its specific role.
And there's actually a drug called tunicamycin
Okay.
that inhibits the very first step in N-linked glycosylation,
Okay.
preventing the transfer of GLCNAC to dolichophosphate phosphate.
So it's like telling the construction worker to take a break,
Yeah,
effectively halting the whole glycosylation process.
exactly.
Okay.
Tunicamycin is a valuable tool for studying the role of N-linked glycosylation in protein function.
It's fascinating how scientists can use these inhibitors to dissect complex cellular processes.
Absolutely.
It's like a molecular toolkit
Yeah.
for probing the inner workings of the cell.
And remember, N-linked glycosylation,
Yeah.
that happens in the ER.
Right.
O-linked glycosylation, which involves adding sugars to serine or threonine residues, that occurs in the Golgi apparatus. And importantly, glycosylation does not happen in the cytosol.
So it's like the cell has designated compartments for these modifications, making sure the right sugars are added in the right place at the right time.
Exactly. It's a testament to that incredible organization and regulation.
Mm-hmm.
Speaking of regulation, protein synthesis doesn't just happen in a vacuum. It's tightly controlled
Right.
by a complex network of signaling pathways and regulatory mechanisms. Cells need to be able to ramp up protein synthesis when demand is high
Okay.
and shut it down when it's not needed.
That makes sense. Wouldn't want to waste energy just churning out proteins if the cell doesn't need them.
Exactly.
So how do cells regulate protein synthesis?
Well, there are multiple levels of regulation.
Yeah, good.
One key level is transcriptional regulation.
Okay.
Controls the rate at which genes are transcribed into mRNA,
Mm-hmm.
involves transcription factors,
Okay.
binding to specific DNA sequences,
Mm-hmm.
and either activating or repressing gene expression.
So it's like a volume knob for each gene,
Yes, that's a good way to think about it.
controlling how much mRNA is produced.
Exactly. And another level of regulation is translational regulation.
Okay.
It controls the rate at which mRNAs are translated into proteins.
Right.
This can involve regulating the initiation of translation, elongation of that polypeptide chain, or even the degradation of the mRNA itself.
So there's a lot of fine-tuning going on
Oh, yeah.
at the molecular level. It's incredible how the cell manages to keep everything in balance.
It is. And there are even regulatory mechanisms that target specific proteins,
Okay.
controlling their activity, their location, or their stability.
Mm-hmm.
For example, a protein might be phosphorylated to activate it, or it might be ubiquitinated to target it for degradation.
So it's not just about making proteins, it's about like managing them
Yeah,
throughout their lifespan.
precisely. This intricate web of regulatory mechanisms ensures that the cell has the right proteins in the right amounts at the right times to carry out its functions.
It's a beautiful system, but incredibly complex. Starting to get a headache just thinking about all the moving parts.
I understand.
A lot to take in.
It is, but I think it's worth the effort.
Mm-hmm.
Understanding these intricacies of protein synthesis gives us a deeper appreciation for the remarkable complexity of life, those elegant mechanisms that cells have evolved to create order out of chaos.
Yeah, it really is a humbling experience to kind of delve into these molecular details and realize how much is going on inside our cells.
It is. Speaking of humbling experiences, our exploration of protein synthesis wouldn't be complete without discussing the role of antibiotics.
Right, right.
These drugs, which have revolutionized medicine,
Yeah.
target specific aspects of bacterial protein synthesis, effectively shutting down bacterial growth, allowing our immune systems to fight off infections.
Yeah, antibiotics are a powerful weapon in our arsenal. But before we get into how they actually work, let's just step back and remind our listeners why bacteria are so vulnerable to these drugs in the first place. Why are their ribosomes, the sites of protein synthesis, such good targets?
That's a great question.
Yeah.
As we discussed earlier, bacterial ribosomes, they're different from eukaryotic ribosomes.
Mhm.
Smaller, simpler,
Right.
have distinct structural features that make them susceptible to certain antibiotics. These antibiotics exploit these differences to selectively inhibit bacterial protein synthesis without harming our own cells.
So like finding a chink in the armor, a vulnerability that we can exploit.
Exactly. And over the years, scientists have developed a wide range of antibiotics that target different steps in bacterial protein synthesis, from initiation to elongation to termination.
It's amazing the power of human ingenuity and the importance of understanding these these molecular details of cellular processes. So let's dive into some specific examples. Can you walk us through how some of these antibiotics actually work?
Sure.
Okay.
Let's start with tetracyclines.
Tetracyclines. Okay.
These antibiotics bind to the 30S subunit of the bacterial ribosome, specifically to the A site, blocking the binding of aminoacyl tRNAs.
So they're like molecular bouncers at the ribosomal nightclub,
Yeah, good analogy.
keeping those aminoacyl tRNAs out.
Exactly. And without those aminoacyl tRNAs, protein synthesis just stops.
Okay, what about aminoglycosides? How do they work?
Aminoglycosides like streptomycin and gentamicin, they also bind to the 30S subunit of the bacterial ribosome.
Okay.
But they have a different mechanism of action. They cause the ribosome to misread the mRNA,
Oh, okay.
leading to the production of faulty proteins.
So like introducing typos into the genetic code.
Exactly. And those faulty proteins, they can't carry out their normal functions, effectively crippling the bacteria.
Okay. What about macrolides? I've heard of those.
Macrolides like erythromycin and azithromycin, they bind to the 50S subunit of the bacterial ribosome,
Okay.
specifically to the peptidyltransferase center, blocking the formation of peptide bonds.
So like little saboteurs cutting the chains that link the amino acids together.
Precisely. Without peptide bond formation, the polypeptide chain can't grow, protein synthesis is halted.
So all these antibiotics, they target these different aspects of protein synthesis, but it all leads to the same outcome, bacterial death.
It does. It really highlights the importance of understanding these molecular details for developing effective therapies.
But there's a catch, right? Antibiotic resistance.
Yeah, unfortunately. The widespread use of antibiotics has led to the emergence of antibiotic-resistant bacteria.
Right.
These bacteria have evolved mechanisms to evade the effects of antibiotics, making them much more difficult to treat.
So it's like an arms race between us and the bacteria.
Exactly, and it's a race that, frankly, we're not always winning.
Yeah.
Antibiotic resistance is a major public health threat, and it's crucial that we develop new strategies to combat it.
So what kind of strategies are scientists exploring?
Well, one strategy is to develop new antibiotics, ones that target different aspects of bacterial physiology,
Okay.
like cell wall synthesis or DNA replication. Another strategy is to develop drugs that block the mechanisms of antibiotic resistance.
So find new ways to outsmart the bacteria.
Exactly. It's a challenge that requires a lot of collaboration between scientists, clinicians, and policymakers.
This whole discussion about antibiotics has been really fascinating, but also a little sobering. It's a reminder that our understanding of protein synthesis has these profound implications for human health.
It does, and it underscores the importance of continued research in this field, not only to combat those bacterial diseases, but also to explore the potential of protein synthesis as a target for other therapies, like anti-cancer drugs.
Oh, wow. So be able to manipulate protein synthesis to treat a wider range of diseases.
Yeah, it's a promising area of research. I'm confident that we'll see some really significant advances in the years to come.
Well, listener, I think we've covered a lot of ground today. Prokaryotic and eukaryotic protein synthesis, explored the fascinating world of post-translational modifications, and even touched on that crucial role of protein synthesis in antibiotic action and resistance.
It's been a whirlwind tour,
Yeah.
but hopefully enlightening. Protein synthesis is a truly remarkable process, a real testament to the elegance and efficiency of cellular mechanisms.
It really is. Just makes you realize how much is going on inside our cells every second of every day.
It does.
And how much we still have to learn.
Always more to discover. That's the beauty of science, right? It's a never-ending quest for knowledge and understanding.
Well, until our next deep dive, keep exploring, keep questioning, keep marveling at the wonders of the microscopic world. It really is a universe unto itself, just filled with endless possibilities.
Indeed. A world that's both familiar and foreign. A place where those building blocks of life are assembled with this exquisite precision, and where that symphony of cellular processes plays out with remarkable harmony.
So, until next time, listener, keep diving deep, and remember, there's a whole universe waiting to be explored, right inside your own cells.